Hello, everyone. Let's look at the Cori cycle. It's something very interesting that's happening in human cells, in animals in general, actually. So, as we said, gluconeogenesis is the making of glucose. That's what it literally means. And originally, it was known to happen in the liver. And in fact, the input here was lactate. So it was not pyruvate. So this is something special, right? So it's a different input. And where does he get lactate from? So muscles make a lot of lactate. So liver doesn't. So it gets the lactate from the muscle. So the muscles, when they work really hard, they do anaerobic glycolysis. So they just do fermentation, right? So they generate a lot of lactate. There is a lactate dehydrogenase, which is a favorable reaction. It takes pyruvate and NADH and generates lactate, and the all-important NAD+, which then can go back into glycolysis in the CAPTase reaction, and glycolysis can keep going. That's the whole point of fermentation, right? To get your ATPs anaerobically just from glycolysis. So, and that's it. That is a favorable reaction. It's not enough to give you an ATP, but as we had calculated, it was about 100 millivolts. The delta E plus 100 millivolts. So it is favorable. Now, so that's fine. That's how it works in muscle, okay? And because muscles have they need to get they need their NAD plus to keep going glycolysis, so they tend to have a low NAD plus to NADH ratio. But in the liver, okay, this lactate dehydrogenase reaction is actually reversed. And so this is how you get pyruvate from lactate. So you get the pyruvate, and then you have to reverse everything else that we discussed previously. You reverse the pyruvate kinase, the PFK step, and so on and so forth. And actually, in the liver, you also reverse the hexokinase step. But how you get pyruvate in the liver, and that's the uniqueness of the Cori cycle, is that you make it from lactate. So you reverse also fermentation in the liver, and that's possible. I just told you, in the muscle, it's thermodynamically favorable based on the delta E, and you get 100 millivolts. But in the liver, you have a very high NAD plus to NADH. So that's why it's possible. So you drive through mass action, essentially. You drive the reverse reaction. And then something really cool happens. So you start in the muscle, okay, you break down glucose, you get your 2 ATPs, and then you do fermentation to generate lactate. The lactate gets out in the circulation, okay, and then it gets picked up by the liver. The liver now, as I said, it can reverse lactate dehydrogenase reaction, give you pyruvate, and then through all the steps we just discussed, okay, it can make glucose, free glucose, okay. Which is energy. I mean, it consumes ATPs for that more than the muscle gains, okay. But because it can make glucose, it can keep feeding glucose to the muscle. So the liver is really your servant there. If you're working really, really hard, it enables the muscle to go for a little longer, okay. To add, the liver adds glucose, therefore adds input into anaerobic glycolysis in the muscle. So this cycling of carbon, essentially, from glucose to lactate was discovered by the Coris, Gerty and Carl Cori. I urge you to look at their history. Here they are at the WashU in St. Louis. Gerty didn't even have a proper tenured faculty position, even though they got the Nobel Prize. I think at that point she got the position, but it was in the 40s and 30s and 40s, and it was very unusual for women to be professors, I suppose, at that point. So lots of discrimination, sex discrimination at the time. Some of it still to this day. All right, so we're going to close with the Cori cycle here, and we're going to finish gluconeogenesis in the next video, where we're going to discuss even more general inputs. How do you make glucose in general? Okay, and we'll stop here.